The land of Iran, known as the Iranian Plateau, has always been a bustling crossroads for people moving across continents, profoundly shaping human history and the spread of populations across Europe and Asia. This region has seen countless civilizations rise and fall, making its genetic story incredibly fascinating. Thanks to new research in ancient DNA, we're now able to uncover the movements of past populations and their genetic ties, giving us unprecedented insights into deep history. By examining genetic material from ancient human remains, scientists can piece together the genetic makeup of past societies, tracing their origins, migrations, and interactions over thousands of years. This powerful method offers a direct genetic window into historical events that were previously only guessed at through archaeology or linguistics. Recent discoveries in archaeogenetics are revealing a truly remarkable and perhaps unexpected story about Persian DNA. Contrary to what many might assume about a region marked by so many historical upheavals and conquests, these studies highlight significant genetic continuity and unique ancestral contributions that have woven the enduring genetic tapestry of the Iranian Plateau. The surprising part lies in the resilience of these ancient lineages and how external influences had a more nuanced impact than previously thought. This persistence challenges common beliefs about how major historical events, like conquests and the rise of vast empires, affect the genetic makeup of populations. The consistent emphasis on 3,000 years of genetic continuity and population stability from prehistoric to modern times. Despite drastic political events and external contacts, truly underscores the remarkable endurance of the underlying genetic heritage. This suggests that big cultural and linguistic changes, like the spread of Indo-Iranian languages, might have happened through things like elite dominance, cultural diffusion, or assimilation, rather than just large-scale population replacement. This gives us a much more intricate understanding of how cultural, linguistic, and genetic histories intertwined in the region. The genetic tale of the Iranian Plateau begins with its very first inhabitants. The earliest human populations in this area, known as Iranian hunter-gatherers, are believed to have originated from the local Upper Paleolithic population. This region is considered a potential birthplace for the deeply diverged Basal Eurasian lineage, an ancient ancestral component that stands out because it has very little to no Neanderthal DNA, setting it apart from other non-African populations. Mesolithic hunter-gatherer remains found in the Alborz Mountains of northern Iran, primarily show ancestry linked to these Basal Eurasians, establishing these early inhabitants as a foundational part of the region's genetic landscape. The shift to agriculture on the Iranian Plateau, a crucial moment in human history, saw a significant contribution from these local populations. Early Neolithic farmers, like those from Ganj Dareh, are thought to be descendants of Iranian hunter-gatherers and are genetically linked to Caucasus hunter-gatherers, CHG. A recently analyzed early Chalcolithic sample from Gol Afshan Tepe, which predates other Chalcolithic genomes from Iran, shows a strong connection to early Neolithic Iranian genetic ancestry. This discovery highlights a long-term cultural and biological continuity in and around the Zagros area, suggesting that the adoption of farming was largely taken up by existing local populations, rather than being brought in by entirely new groups. While local continuity was a dominant theme, early periods also show some evidence of genetic influence from the West. The genetic profiles of historical samples from northern Iran appear as intermediates on an east-west genetic gradient across the Persian Plateau, indicating ongoing interactions and subtle gene flow from neighboring regions. This suggests a dynamic yet integrated genetic landscape from early times, where local populations maintained their core genetic identity while absorbing some external genetic components. The Iranian Plateau was more than just a passageway. It was a primary source of early Eurasian genetic diversity. Ancient DNA studies reveal that the region acted as a population hub during the initial colonization of Eurasia after the out-of-Africa expansion. Furthermore, Iranian hunter-gatherers and Neolithic groups hold an extreme position in genetic analyses compared to other West-Eurasians, and their ancestry was initially absent from Anatolia, Mesopotamia, the Caucasus, and India, only reaching these regions later through the expansion of Mesolithic and Neolithic groups. This implies that the Iranian Plateau wasn't just a passive recipient or a bridge for migrations, but an active contributor to the genetic makeup of surrounding areas, including parts of the Near East and possibly South Asia. This perspective redefines the Iranian Plateau's historical importance, emphasizing its foundational contribution to the genetic tapestry of a much broader geographical area. The genetic makeup of prehistoric groups on the Persian Plateau formed part of a genetic gradient that stretched across the entire region from east to west. This broad genetic pattern, showing gradual genetic variation across the landscape, continued into the historical era, indicating persistent, though regionally varied, connections across the vast plateau. Such a gradient suggests a long history of interconnectedness and gene flow throughout the region, rather than sharp genetic boundaries. Historical samples from the northern Iranian Plateau show strong genetic links to local and South-Central Asian Bronze Age populations, particularly those of the Bactria-Margiana Archaeological Complex, BMAC, or Oxus civilization. The ancestries of these northern Iranian groups can largely be represented by the peoples of BMAC and their descendants, highlighting long-term genetic ties across these regions. This enduring genetic connection further expanded under the First Persian Empire, the Achaemenids, whose core territories included the Iranian Plateau, South Caucasus, South-Central Asia, and parts of modern Afghanistan and Pakistan. Mesolithic Caucasus hunter-gatherers, CHG, and Iranian Neolithic farmers significantly contributed to later groups in the region. In various parts of the plateau, studies describe dual ancestries, originating from both CHG and Iranian farmer populations, indicating complex admixture patterns. CHG groups are also very closely related to Mesolithic and Neolithic populations on the Iranian Plateau, sometimes even being included within the broader CHG Iranian hunter-gatherer cluster. This demonstrates a shared deep history and ongoing genetic exchange between these adjacent regions. The persistent east-west genetic gradient and the enduring genetic connections across these regions, especially with South-Central Asia, imply that long-standing trade and cultural exchange routes such as the Silk Road were not merely economic or cultural arteries, but also pathways for gene flow. By the 1st century BCE, the Persian Plateau had become a crucial bridge between the East and the West, through extensive trade and cultural exchange. This suggests that these historical networks played a subtle yet significant role in shaping the genetic landscape over extended periods, reflecting a more integrated Eurasian network than typically perceived. The movement of goods and ideas along these routes was accompanied by a gradual, continuous exchange of genetic material, contributing to the observed genetic patterns across the plateau and its neighboring regions. A central finding in recent ancient DNA studies is the substantial genetic continuity observed in the northern Iranian Plateau, spanning approximately 3,000 years from the Copper Age to the Sassanid Empire, 4700 BCE to 1300 CE. This remarkable stability persists despite the rise and fall of major empires and significant cultural and political transformations that reshaped the region's geopolitical landscape. This continuity is evident across various time periods and archaeological sites, providing a robust picture of population persistence. The genetic profiles of historical samples, especially from the Achaemenid, Parthian, and Sassanid periods, 355 BCE, 460 CE, show strong connections to local and South-Central Asian Bronze Age populations. These individuals are positioned as intermediates on the east-west genetic gradient across the Persian Plateau, reflecting a consistent underlying genetic structure that endured through these imperial eras. This indicates that the core population of the Iranian Plateau maintained its genetic identity even as it became part of vast political entities. Analyses of uniparental lineages, Y-chromosomal DNA for paternal lines and mitochondrial DNA for maternal lines, on the Iranian Plateau further highlight this population stability. Many such lineages were found to be continuous over centuries and are still present in modern groups of the region, providing a direct genetic link across vast spans of time. This persistence of specific genetic markers across millennia offers compelling evidence for the enduring nature of the local gene pool. The coexistence of such profound genetic continuity with drastic political events and external contacts during the periods of the Achaemenid, Parthian, and Sassanid empires suggests a nuanced impact of imperial rule on human populations. The fact that genetic profiles during these vast empires show strong connections to local and South Central Asian Bronze Age populations, rather than significant genetic replacement, implies that these empires might have functioned more as integrators of diverse existing populations and facilitators of cultural diffusion, rather than agents of massive population movements that fundamentally altered the genetic makeup of the core regions. This offers a more subtle view of imperial impact on human genetics, where political dominance did not necessarily equate to demographic upheaval. The spread of Indo-Iranian languages into the Iranian Plateau, closely related to Indo-Aryan languages, is traditionally linked to migrations from the Central Asian steppes, specifically associated with the Sintashta and Andronovo cultures, after 2000 BCE. This linguistic shift has often been associated with significant genetic contributions from these steppe pastoralist groups, implying a demographic replacement or substantial admixture accompanying the language spread. However, a significant and surprising finding from recent ancient DNA studies is the notable absence or limited detectable admixture from Bronze Age steppe pastoralists in key historical northern Iranian groups, particularly those from the Achaemenid, Parthian, and Sassanid periods. Instead, these populations exhibit strong genetic connections to local and South Central Asian Bronze Age populations, such as those associated with the Bactria-Margiana Archaeological Complex, BMAC, while some modern Iranian populations show a general presence of Steppe ancestry, estimated around 20%. The specific ancient DNA analysis for historical northern Iranian individuals indicates a different pattern for the core historical periods. This observation is particularly compelling as it challenges a direct one-to-one correlation between linguistic diffusion and large-scale genetic influx. This finding carries important implications for understanding population movements in the region. It suggests that the spread of Indo-Iranian languages and culture into the Iranian Plateau may have occurred through mechanisms other than large-scale genetic replacement, such as cultural diffusion, elite migration, or language shift among existing populations, where a smaller group introduces a language that is then adopted by a larger indigenous population. This contrasts with the significant infusion of male steppe ancestry observed in parts of the Indian subcontinent, highlighting regional differences in the genetic impact of Indo-European expansions. The observation that historical northern Iranian individuals did not show detectable admixture from Bronze Age steppe pastoralists, despite the linguistic evidence for Indo-Iranian presence, is critical for refining models of language dispersal. It suggests that the adoption of new languages, even those associated with powerful migrating groups, does not necessarily require a large-scale genetic influx. This opens up avenues for understanding cultural and linguistic transmission through more complex social dynamics, such as elite emulation or cultural prestige, rather than solely demographic shifts. Modern Iranian populations display a diverse array of genetic lineages, reflecting their complex history as a crossroads of human migration and interaction. Large-scale assessments identify common West-Eurasian mitochondrial DNA, mtDNA, haplogroups such as U, H, J, HV, despite Iran's ethnic diversity and significant geographical barriers like the Zagros and Alborz mountain ranges and the Dasht-e-Kavir and Dasht-e-Lut deserts. Studies show that language plays a crucial role in shaping the modern Iranian gene pool. In some analyses, linguistic grouping explains a higher percentage of genetic variation among Iranian groups, 2.69%, than geography, 2.18%, or ethnicity, 2.03%. This suggests that shared linguistic identity can correlate strongly with genetic clustering, indicating historical processes where language boundaries may have also served as barriers or facilitators of gene flow. Genetically, Iranian peoples, including the Persian-speaking majority and significant minorities such as Azeris, Kurds, Lurs, and Balochis, tend to cluster tightly together, forming a Central Iranian cluster, CIC. This cluster positions them closely with other West Asian and European populations, reinforcing their predominantly West-Eurasian genetic affinity. Interestingly, within this broader cluster, linguistic identity is sometimes less relevant than this overarching genetic similarity. For example, Iranian Arabs and Azeris can be nearly indistinguishable from other Iranian groups genetically, despite speaking different language families. The complex interplay observed between language, ethnicity, and genetics in modern Iran reveals a nuanced picture. While language emerges as a strong differentiator when analyzing the overall variation among Iranian groups, there is a surprising degree of genetic homogeneity within the Central Iranian cluster, despite the linguistic diversity. This indicates that historical processes, including language shifts without full population replacement, have created a situation where distinct linguistic groups can share a common, deeply rooted genetic heritage, particularly within the core Iranian Plateau. This understanding underscores that genetic ancestry, linguistic affiliation, and ethnic identity are not always perfectly aligned, highlighting the multifaceted nature of human population history. The comprehensive analysis of ancient and modern DNA reveals that the genetic origins of Persian populations are characterized by a profound and enduring continuity. This continuity is deeply rooted in indigenous hunter-gatherer and early Neolithic farmer ancestries that have persisted for millennia. This long-term stability, spanning approximately 3,000 years from the Copper Age to the Sassanid Empire and extending into modern times, stands as a truly remarkable aspect of the region's genetic history, challenging assumptions of widespread demographic upheaval, despite numerous political and cultural transformations. The Iranian Plateau has served not only as a crucial geographical bridge for human movements between East and West, but also as a significant genetic reservoir. It has contributed unique, ancestral components to the broader Eurasian landscape, demonstrating its role as an active source of genetic diversity rather than merely a passive corridor. The genetic tapestry of the region reflects a dynamic interplay of local persistence and gradual, integrated external influences shaped by long-standing interactions and trade networks. Furthermore, the evidence of limited detectable Bronze Age steppe pastoralist admixture in historical northern Iranian populations refines simplistic models of language and cultural diffusion. This finding suggests that the spread of Indo-Iranian languages into the plateau may have been more complex than previously assumed, potentially involving cultural adoption or elite migration rather than extensive demographic replacement. This offers a more nuanced understanding of how linguistic and cultural shifts can occur independently of major genetic shifts. While current archaeogenetic studies have provided groundbreaking insights into the genetic narrative of the Persian Plateau, the field continues to evolve. Future research, particularly with broader geographical sampling across the entire Iranian Plateau and more detailed chronological resolution, will further enhance our understanding of regional variations, the precise nature of early genetic components, and the long-term impacts of various historical contacts. These ongoing investigations promise to unveil even more layers of the rich and stable genetic heritage that defines the populations of the Iranian Plateau.